In this segment, we will discuss about an important technique of molecular biology or recombinant DNA technology, that is PCR, polymerase chain reaction. Right? So PCR is called as, the full form is polymerase chain reaction, that is PCR. This technique was first given by Carey B. Mullis in the year 1985. So first given by Carey B. Mullis in the year 1985, for which was rewarded with the Nobel Prize in the year 1993. And the Nobel that he received is in the chemistry. So first given by Carey B. Mullis in the year 1985 and rewarded Nobel Prize in the chemistry in the year 1993. This technique is used for DNA amplification. Used for amplification of DNA. Amplification means increase the copies, increase the number of copies. Now this technique has a major role. Say for example, when we have obtained DNA from any criminal scene or the crime site. Also we know that the size of the DNA sample that you will receive, say for example a simple or a single smear of the blood, a simple blood drop we are receiving. So from that blood drop we know that RBCs are present, WBCs are present and platelets are present. Platelets, we will not get any DNA. From RBCs also we are not going to get any DNA. So the DNA that we are getting is only and only from WBCs, and we know the normal value of the WBC is 6,000 to 10,000 or 11,000 per cubic mm of blood. So from that small size of the small number of the WBC, we need only small sample of the DNA. And say for example, if anything undesirable things happen throughout the experimental period of time, let us say or let us assume that the sample that we was having, initial sample, has gone. Now it is lost. Now we don't have any sample. So how we will trace the criminal? So for that reason, we need the extra copies of the DNA. We need to amplify the DNA, and the amplification technique is used is polymerase chain reaction. This PCR is completed in the 3 steps. Right? The 3 steps of the PCR are denaturation, annealing and extension. Denaturation, annealing and extension. Right? So let us discuss these 3 steps one by one. So first is denaturation. Denaturation is basically breakdown of the hydrogen bonds between the adjacent base pair and convert the double-stranded DNA into single-stranded DNA. So this denaturation is basically breakdown of hydrogen bonds between adjacent base pairs. Right? We know adenine binds with the thymine via 2 hydrogen bonds, guanine binds with the cytosine via 3 hydrogen bonds. That we know. So say for example, if these 2 are the strands, that is the parent DNA or original DNA copy that we are talking of, then denaturation is the first step in which the hydrogen bonds between the 2 strands, purines and pyrimidines, will break, and now what is we are going to obtain is the initial one is this DNA. Right? Now what we will get is the 2 strands. Each of the strand will now act, we know it is a template DNA. Each strand will act as a template DNA or a template strand. Now This template strand is acting as a kind of copy, a template, and based on the complementarity sequence, new nucleotides will be added in the subsequent steps. So denaturation, it is a breakdown of hydrogen bonds between the adjacent base pairs. Adenine binds with the thymine via 2 hydrogen bonds. Guanine pairs are bind with the cytosine via 3 hydrogen bonds. Right? Initially it is a double-stranded DNA, dsDNA. Now it is a single-stranded DNA. Both the DNAs will act as a template DNA strand, and based on the complementarity, the new nucleotide will be added. Denaturing agents, acids can be used, bases are also used, but specifically in the polymerase chain reaction, the denaturing agent or the source by which denaturation is done is temperature. So very high temperature of 90 to 94 degrees Celsius is the range of the temperature, and the optimum temperature is 92 degrees Celsius. Right? So this is the first step, that is what we call it as denaturation. Now the second step is annealing. Annealing basically means hybridization or the attachment. Attachment of what? We have discussed in detail the mechanism of DNA replication. We know that the primers are required, right, for DNA replication to begin. One more thing, 90 to 94 degrees Celsius or the 92 degrees Celsius, which is the optimum temperature, the DNA is heated only for 15 seconds. Right? We are not breaking the phosphodiester bonds, we are breaking only hydrogen bonds. So, for a very short duration of time. Annealing is basically attachment or hybridization of what? Now primers are going to add onto the template DNA. Primers are added onto the template DNA. Right? Now again, the location of the primer is very, very important. This is the 5' end of the DNA, 3' end. This is 5' end, 3' end. We know that the DNA synthesis, new DNA or nucleic acid DNA or RNA synthesis begins towards 3' OH. Right? Because 3' OH is basically acting as a functional group. New nucleotide can come and join at the area or the site where the functional groups are located. And functional group is located at only one end, that is 3' OH. 5' end is free phosphate, 3' end is free OH. So let us assume if the primer attaches here, right? So this would be the 3' end of the primer, this would be the 5' end of the primer. So whether this is correct, this is not correct. Let us understand this sequence. 3' is the parent strand and the 5'. 3' to 5', this is the parent strand, right? Let us say the primer attaches here, then this would be the 5' end of the primer and this would be the 3' end of the primer, right? And we know 3' end is free OH group, so new nucleotide can come and bind. So this is the correct position of the primer towards the 3'. So new nucleotide will come and join, right? So parent strand is 3' to 5'. Primer is 5' to 3' because it is towards the 3' OH direction. So in this case, primer has to be attached here. At this side. Because if this is 3', this would act as a 5' of the primer. And this will be the 3' end of the primer. Right? Now the primer has been attached. Primer is basically a short oligonucleotide sequence of RNA. Right? Primer is short oligonucleotide sequence of RNA, approximately 8 to 10 nucleotide long. In many reference book it is written 10 to 18 also, but generally it is 8 to 10 nucleotide sequence which is involved as a part of primer. Now this temperature, the annealing basically takes place at a lower temperature. The temperature that is required is 50 to 60 degrees Celsius. That is the range of the temperature, and the temperature optimum is 55, or you can say 56 degree Celsius is the optimum temperature range where annealing is carried out. Right? So denaturation, we have studied annealing. Then the third step is extension or elongation. So denaturation done, annealing done, now extension or elongation. Anyone you can refer. Extension or elongation. So basically, now what we have in our hands, we have the template DNA, we have the primer, right? What we are waiting for? We are waiting for only an enzyme, that is DNA polymerase, because DNA polymerase recognizes the override sequence, that is origin of replication sequence. Primer attaches, new nucleotide formation takes place. So what we require is, we require enzyme DNA polymerase. We also require nucleotides, that is the raw material. Nucleotides, which is basically acting as a raw material for the synthesis of new DNA strand. Now talking about this DNA polymerase. The DNA polymerase that is acting or functional within the body of human eukaryotic human cell is activated at 37 degrees Celsius only. But here, are we using 37 degrees Celsius? No. The first step, that is denaturation, takes place at the 92 degrees Celsius, right? Annealing takes place at about 55 or 56 degrees Celsius. So it is beyond 37 degrees Celsius, which is the normal acting or the optimum temperature of DNA polymerase. But in this case, we have to use a thermostable DNA polymerase. We have to use thermostable DNA, all that is polymerase. The thermostable DNA polymerase is called as tag polymerase, which is isolated from Thermus aquaticus bacterium. So name of the polymerase is tag polymerase. This tag polymerase is isolated from Thermus aquaticus. From Thermus aquaticus bacterium, which is basically a typical archaebacterium, and they grows in hot water springs. So if they are growing, this bacterium is growing in the hot water springs, that means the bacterium can tolerate the high temperature. So whatever enzyme, whatever other molecules that the bacterium is producing can withstand higher temperatures, like beyond 90 degrees Celsius also. So tag polymerase is the name of the bacterium, name of the enzyme, sorry. It is isolated from Thermus aquaticus bacterium, which is a typical archaebacterium, can grow in the hot water springs. This tag polymerase is thermostable enzyme, right? So nucleotides are also added. DNA polymerase, that is tag polymerase, is nothing, it is a DNA polymerase only, but a modified version, right? Tag polymerase, that is DNA polymerase we have, raw material, that is nucleotide we have. Now extension or elongation will take place. One more example that I can discuss with you is Vent polymerase. This is the tag polymerase generally used in PCR. Other enzyme, that is Vent polymerase. This Vent polymerase is isolated from Thermococcus litoralis, name of the bacterium. It is isolated from Thermococcus litoralis, right? So denaturation, annealing as well as extension. We started with the one DNA. Now what we will get is template is there, nucleotides are also there, DNA polymerase is also there. Now what we will get? One parent strand and one daughter strand. means one old strand we will get and one new strand we will get. So from one we have got 2 DNA strands, right? We will not stop it here only. We have to go for the continuous cycles of PCR, 27 cycles, 28 cycles, 25 cycles, 30 cycles to reach up to the 1 million copies of DNA. These 2 copies of the DNA we obtain. Again we can send to denaturation. Again same steps will take place. Again after denaturation and annealing, that is primer will be added. After annealing, elongation. So from 2 we will get 4 like that. Okay. So this is all about polymerase chain reaction, that is PCR, an important technique